Today we will discuss about CAM pathway. In last lecture we discussed about the C4 plants. Let's start CAM pathway. CAM stand for crassulacean acid metabolism, and this is a photosynthetic adaptation in succulent plants. Succulent plants, also known as fat plants, are xerophytic plants adapted to arid climates or soil conditions. Succulent plants store water in their leaves and stem. The storage of water often gives succulent plants a swollen or fleshy appearance than other plants, and this characteristics is known as succulence. If we talk about the example of succulents, best example is cactus. These xerophytic plants open their stomata during the night and close their stomata during day. This closing stomata during the day helps succulent plants conserve water, but it also prevents entering of CO2 from leaves. During night, when their stomata are open, these plants take up CO2. Assimilation of CO2 occurs into malic acid at night, which is stored in the vacuole. This mode of carbon fixation is called crassulacean acid metabolism or CAM. After the plant family Crassulaceae, the succulent in which the process was first discovered. So during the daytime, when the light reactions can supply ATP and NADPH for the Calvin cycle, CO2 is released from the malate for fixation through Calvin cycle. This cycle differs from the C4 cycle. In C4 plants, formation of the malic acid in the mesophyll is spatially separated from the Calvin cycle in the bundle sheath. In CAM plants, the two steps occur at separate time. Here, the pathway for CAM plants shown in figure. Characteristics of these plants: that in dark their stomata open and in light their stomata closed. When stomata open, stomata permits entry of CO2 and loss of water. So finally, during dark, stomata open and CO2 enters into plants. During night, closed stomata prevent entry of CO2 into plants. At night, CO2 is captured by PEP carboxylase. CO2 is captured by PEP carboxylase in the cytosol of mesophyll cell, and PEP is carboxylated to oxaloacetate. Then PEP is converted to oxaloacetate. The oxaloacetate is reduced to malate, which is finally stored in the vacuole. Oxaloacetate is converted into malate and stored in the vacuole. Reduction of oxaloacetate is catalyzed by enzyme which is known as NAD+ malate dehydrogenase. If we talk about during the day, during daytime, the stored malate is transported back into the cytosol. This stored malic acid, which is stored in vacuole, is transported back into the cytosol, where it is decarboxylated by a NADP+ malic enzyme. The CO2 just released enters the chloroplast, where it is fixed by the Calvin cycle. The Adaptive advantage of CAM is the reduction of water loss by transpiration achieved by the stomatal opening during night. This is all about the CAM pathway. Let's discuss about the differences between C3 plants, C4 plants and CAM plants. In last lecture we discussed about C4 plants. Last lecture we discussed about the C3 plant. There are some differences between the C3, C4 and CAM plants. Let's discuss. First difference is, if we talk about the Crange anatomy, which we discuss about the about in C4 plants. Crange anatomy in C3 plant, this type of anatomy is absent. In C4 plant, this Crange anatomy is present, and in CAM plants also this is absent. Now second difference is carboxylating enzyme. In C3 plant, RUBP carboxylase act as carboxylating enzyme. In C4 plant, in mesophyll cells, PEP carboxylase, and in bundle sheath cell, RUBP carboxylase act as carboxylating enzyme. And in CAM plants, in dark means in night, PEP carboxylase, and when light is present, RUP carboxylase act as carboxylating enzyme. Next difference is initial CO2 acceptor, who act as first acceptor of CO2. In C3 plant, RUBP act as CO2 acceptor, first CO2 acceptor. In C4 plants, PEP, phosphoenol pyruvate, act as CO2 acceptor, and in CAM plants also phosphoenol pyruvate act as initial CO2 acceptor. Next difference is first stable product. First stable product formed in C3 plant is 3-PZA, which was a 3-carbon product. In C4 plants, first stable product is oxaloacetate, and in CAM plants, first stable product is also oxaloacetate. These both are in C4 plants, and in CAM plants, first stable product is 4-carbon compound. Next difference is optimum temperature for photosynthesis. For C3 plants, optimum temperature for photosynthesis is 15 to 25 degrees Celsius. In C4 plants, this optimum temperature is 30 to 40 degree Celsius. And in CAM plants, this optimum temperature is less than 40 degree Celsius. Next difference is CO2 compensation point. For C3 plants, CO2 compensation point is 30 to 70 ppm. For C4 plant, it is 0 to 10 ppm, and in CAM plant, this is 0 to 5 during night. Next difference is photorespiration. In C3 plant, photorespiration is present. In C4 plant, photorespiration is not easily detectable, and in CAM plant, photorespiration is detectable in afternoon. Next difference is Calvin cycle operative. In C3 plant, Calvin cycle operated alone. In C4 plants, Calvin cycle operated along with the H and Slack cycle, and in CAM plant, Calvin cycle operated along with the H and Slack cycle. These all are some differences between C3, C4 and CAM plants.